Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Prokaryotic cytoskeleton. 13 languages. Elements of the Caulobacter crescentus. Cytoskeleton. The prokaryotic cytoskeletal elements are matched with their eukaryotic homologue and hypothesized cellular function. 1. The prokaryotic cytoskeleton is the collective name for all structural filaments in prokaryotes. It was once thought that prokaryotic cells did not possess cytoskeletons, but advances in visualization, technology and structure determination led to the discovery of filaments in these cells in the early 1990s. 2. Not only have analogues for all major cytoskeletal proteins in eukaryotes been found in prokaryotes, cytoskeletal proteins with no known eukaryotic homologues have also been discovered. 3, 4, 5, 6. Cytoskeletal elements play essential roles in cell division, protection, shape determination, and polarity determination in various prokaryotes. 7, 8. Tubulin superfamily, edit. FTSZ, edit. Main article, FTSZ. FTSZ, the first identified prokaryotic cytoskeletal element, forms a filamentous ring structure located in the middle of the cell called the Z-ring that constricts during cell division, similar to the actin myosin contractile ring in eukaryotes. 2. The Z-ring is a highly dynamic structure that consists of numerous bundles of protofilaments that extend and shrink, although the mechanism behind Z-ring contraction and the number of protofilaments involved are unclear. 1. FTSZ acts as an organizer protein and is required for cell division. It is the first component of the septum during cytokinesis, and it recruits all other known cell division proteins to the division site. 9. Despite this functional similarity to actin, FTSZ is homologous to eukaryotic tubulin. Although comparison of the primary structures of FTSZ and tubulin reveal a weak relationship, their three-dimensional structures are remarkably similar. Furthermore, like tubulin, monomeric FTSZ is bound. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF to GTP and polymerizes with other FTSZ monomers with the hydrolysis of GTP in a mechanism similar to tubulin dimerization. 10. Since FTSZ is essential for cell division in bacteria, this protein is a target for the design of new antibiotics. 11. There currently exist several models and mechanisms that regulate Z-ring formation, but these mechanisms depend on the species. Several rod-shaped species, including Escherichia coli and Caulobacter crescentus, use one or more inhibitors of FTSZ assembly that form a bipolar gradient in the cell, enhancing polymerization of FTSZ at the cell center. 12. One of these gradient-forming systems consists of min-CDE proteins, see below. Actin superfamily, edit. MREB, edit. Main article, MREB. MREB is a bacterial protein believed to be homologous to eukaryotic actin. MREB and actin have a weak primary structure match, but are very similar in terms of 3D structure and filament polymerization. Almost all non-spherical bacteria rely on MREB to determine their shape. MREB assembles into a Helical network of filamentous structures just under the cytoplasmic membrane, covering the whole length of the cell. 13. MREB determines cell shape by mediating the position and activity of enzymes that synthesize peptidoglycan and by acting as a rigid filament under the cell membrane that exerts outward pressure to sculpt and bolster the cell. 1. MREB condenses from its normal helical network and forms a tight ring at the septum in Caulobacter crescentus right before cell division, a mechanism that is believed to help locate its off-center septum. 14. MREB is also important for polarity determination in polar bacteria, as it is responsible for the correct positioning of at least 4 different polar proteins in C. crescentus. 14. PAR-M and SOP-A, edit. Main article, PAR-M. PAR-M is a cytoskeletal element that possesses a similar structure to actin, although it behaves functionally like tubulin. Further, it polymerizes bidirectionally and it exhibits dynamic instability, which are both behaviors characteristic of tubulin polymerization. 4. 15. It forms a system with PAR and PARC that is responsible for R1 plasmid separation. PAR-M affixes to PAR, a DNA binding protein that specifically binds to 10 direct repeats in the PARC region on the R1 plasmid. This binding occurs on both ends of the PAR-M filament. This filament is then extended, separating the plasmids. 16. The system is analogous to eukaryotic chromosome segregation as PAR-M acts like eukaryotic tubulin in the mitotic spindle, PAR acts like the kinetochore complex, and PARC acts like the centromere of the chromosome. 17. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. F-plasmid segregation occurs in a similar system where SOP-A acts as the cytoskeletal filament and SOP-B binds to the SOP-C sequence in the F-plasmid, like the kinetochore and centromere, respectively. 17. Lately an actin-like PAR-M homologue has been found in a gram-positive bacterium, Bacillus thuringiensis, which assembles into a microtubule-like structure and is involved in plasmid segregation. 18. Archaeal actin, edit. Crinactin is an actin homologue unique to the archaeal kingdom Thermoproteata, formerly Crinarchaeota, that has been found in the orders Thermoproteales and Candidatus. Coraceum. 19. At the time of its discovery in 2009, it has the highest sequence similarity to eukaryotic actins of any known actin homologue. 20. Crinactin has been well characterized in Pyriobaculum calidifontis, a 3MWN5, and shown to have high specificity for ATP and GTP. 19. Species containing crinactin are all rod or needle-shaped. In P. calidifontis, crinactin has been shown to form helical structures that span the length of the cell, suggesting a role for crinactin in shape determination similar to that of MREB in other prokaryotes. 19. 21. Even closer to the eukaryotic actin system is found in the proposed superphylum of Ascardichaeota. They use primitive versions of profilin, chelsolin, and cafilin to regulate the cytoskeleton. 22. Unique groups, edit. Crescentin, edit. Main article, Crescentin. Crescentin, encoded by crescent gene, is an analog of eukaryotic intermediate filaments, IFs. Unlike the other analogous relationships discussed here, crescentin has a rather large primary Homology with if proteins. In addition to three-dimensional similarity, the sequence of crescent has a 25% identity match and 40% similarity to cytokeratin 19 and a 24% identity match and 40% similarity to nuclear lamina. Furthermore, crescentin filaments are roughly 10 nm in diameter and thus fall within diameter range for eukaryotes, 8 to 15 nm. 23. Crescentin forms a continuous filament from pole to pole alongside the inner, concave side of the crescent-shaped bacterium. Caulobacter crescentus. Both MREB and crescentin are necessary for C. crescentus to exist in its characteristic shape. It is believed that MREB molds the cell into a rod shape and crescentin bends this shape into a crescent. 1. Min-CDE system, edit. Main article, Min system. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The Min-CDE system is a filament system that properly positions the septum in the middle of the cell. In Escherichia coli. According to Shi et al., Min-C inhibits the formation of the septum by prohibiting the polymerization of the Z-ring. Min-C, MIND, and MINE form a helix structure that winds around the cell and is bound to the membrane by MIND. The Min-CDE helix occupies a pole and terminates in a filamentous structure called the E-ring made of MINE at the middlemost edge of the polar zone. From this configuration, the E-ring will contract and move toward that pole, disassembling the Min-CDE helix as it moves along. Concomitantly, the disassembled fragments will reassemble at the opposite polar end, reforming the Min-CDE coil on the opposite pole while the current Min-CDE helix is broken down. This process then repeats, with the Min-CDE helix oscillating from pole to pole. This oscillation occurs repeatedly during the cell cycle, thereby keeping Min-C and its septum inhibiting effect at a lower time averaged concentration at the middle of the cell than at the ends of the cell. 24. The dynamic behavior of the Min proteins has been reconstituted in vitro using an artificial lipid bilayer as mimic for the cell membrane. MINE and MIND self-organized into parallel and spiral protein waves by reaction diffusion-like mechanism. 25. Bactophilin, edit. Bactophilin, Interpro, IPR 007607, is a helical cytoskeletal element that forms filaments throughout the cells of the rod-shaped Proteobacterium Myxococcus xanthus. 26. The bactophilin protein, BAC-M, is required for proper cell shape maintenance and cell wall integrity. M. xanthus cells lacking BAC-M have a deformed morphology characterized by a bent cell body, and BAC-M mutants have decreased resistance to antibiotics targeting the bacterial cell wall. M. xanthus BAC-M protein is cleaved from its full-length form to allow polymerization. Bactophilins have been implicated in cell shape regulation in other bacteria, including curvature of Proteus mirabilis cells. 27. Stalk formation by Caulobacter crescentus. 28. And helical shape of Helicobacter pylori. 29. See also, edit. Cell division. Cyanobacterial morphology. Cytokinesis. Cytoskeleton. Prokaryotes. Protein filament. References, edit. 1 ABCDGTIZ, March 2005. The New Bacterial Cell Biology, Moving Parts and Subcellular Architecture. Cell 120, 557786 do, 10.1016/j.cell.2005.02.026. PMID 157665222. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. S2 SID 88943004. 2-AB by F. Lutkenhorst J. November 1991. FTSZ ring structure associated with division in Escherichia coli. Nature. 354, 6349 1614. Bibcode 1991 nature.354161b. Do 10.1038/35416110. PMID 1944597. S2 SID 43299947. 3 Gunning PW, Ghosh Dastida U, Whitaker S, Pop D, Robinson RC, June 2015. The Evolution of Compositionally and Functionally Distinct Actin Filaments. Journal of Cell Science. 128, 11/2009-19. Do 10.1242/JCS.165563. PMID 257888699. 4-AB Pop D, Narita Lee LJ, Ghosh Dastida U, Xu B, Srinivasan R, Balaju Brahmanian MK, Tanakati. Robinson RC, June 2012. Novel actin-like filament structure from Clostridium tetani. The Journal of Biological Chemistry. 287, 25-211-219 do, 10.1074/jbc.m112.341016. PMC 3375535. PMID 225142779. 5 Pop D, Narita Ghosh Dastida U, Maida K, Maida Y, Adati, Fujisawa Onishi H, Ito K, Robinson. RC, April 2010. Polymeric Structures and Dynamic Properties of the Bacterial Actin Alpha. Journal of Molecular Biology. 397, 103141 do, 10.1016/j.jmb.2010.02.010. PMID 201564449. 6 Wickstead B, Gull K, August 2011. The Evolution of the Cytoskeleton. The Journal of Cell Biology. 194, 451325 do, 10.1083/jcb.2011020065. PMC 3160578. PMID 218598959. 7 Shiwy L, Rothfield L, September 2006. The Bacterial Cytoskeleton. Microbiology and Molecular Biology Reviews. 70, 372954 do, 10.1011.mmBr.00017-06. PMC 1594594. PMID 1695997. 8 Michi K.A., Low J., 2006. Dynamic Filaments of the Bacterial Cytoskeleton, PDF. Annual Review of Biochemistry. 75, 46792. Do 10.1011.biochem.75.1030004.14245 PMID 167564999. Archived from the original PDF on the 17th of November 2006. 9 Grauman P.L., December 2004. Cytoskeletal elements in bacteria. Current opinion in microbiology. 7, 65671 do. 10.1016/j.mib.2004.10.010. PMID 1555627. 10 Daysire, Mitchison T.J., July 1998. Tubulin and FTSZ structures, functional and therapeutic implications. Bioassays. 20, 75237 do. 10.1002/CISI, 1521-1878, 199807, 20:73.0.co, 2L. PMID 9722999. 11 Haydon D.J., Stokes N.R., U.R., Galbraith G., Bennett J.M., Brown D.R., Baker P.J., Bainin V.V., Rice D.W. Sedelnikova S.E., Heel Jr., Sheridan J.M., A. Whale S.T., Chauhan Park, Srivastavara, Tainajuru, Collins I. Errington J., Chapluski L.G., September 2008. An inhibitor of FTSZ with potent and selective anti-staphylococcal activity. Science. 321, 5896 1673 5. Bib code, 2008 SCI, 321.1673H. Do. 10.1026/science.11599961. PMID 188019997. S2 SID 78788853. 12 Hassa DP, Margolin W., April 2016. Splitsville, Structural and Functional Insights into the Dynamic Bacterial Z-ring. Nature Reviews. Microbiology. 14, 530519 do. 10.1038/micro.2016.26. PMC 5290750. PMID 2704057. 13 Kerner J., Medalia O., Lenaraudiz A., Baumeister W., November 2004. New insights into the structural organization of eukaryotic and prokaryotic cytoskeletons using cryo-electron tomography. Experimental cell research. 301, 13842 do. 10.1016/j.yeska.2004.008.005. PMID 155014443. 14 AB Jitaiz, Diane, Shapiro L., June 2004. An actin-like gene can determine cell polarity in bacteria. Proceedings of the National Academy of Sciences of the United States of America. 101. 23643 do. 10.1073/nas.0402638101. PMC 423248. PMID 1515957. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 15 Garner EC, Campbell CS, Mullis Road, November 2004. Dynamic instability in aDNA segregating. Prokaryotic actin homologue. Science. 306, 5698 1021 5. Bibcode, 2004 SCI, 306.1021G. Do. 10.1126/science.1101311313. PMID 155284442. S2 SID 140322209. 16 Mla Jensen J, Jensen RB, Low J, Gerds K, June 2002. Prokaryotic DNA segregation by an actin-like filament. The EMBO Journal. 21, 12 3119 27 do. 10.1093/embodge/CDF 320. PMC 126073. PMID 12065424. 17 AB JTIZ, February 2006. Plasmid segregation, a new class of cytoskeletal proteins emerges. Current biology. 16, 4R133-6. Bibcode, 2006 CBio, 16.R133G. Do. 10.1016/J.Cub.2006.02.007. PMID 1648885. 18 Gian Guess, Narita, Pop D, Ghosh Dastida U, Li LJ, Srinivasan R, Balaju Brahmanian MK, Adati, Co. F, Larson M, Robinson RC, March 2016. Novel actin filaments from Bacillus thuringiensis form. Nanotubules for plasmid DNA segregation. Proceedings of the National Academy of Sciences of the United States of America. 113, 9E1200-5. Bibcode, 2016 NAS 113E1200J. Do. 10.1073/NAS.1600129113. PMC 47806441. PMID 268731005. 19 ABC Etima TJ, Lindas AC, Bernanda R, May 2011. An actin-based cytoskeleton in archaea. Molecular microbiology. 80, 105261 do. 10.1011/J.1035-2065-2011.07635.x. PMID 21414041. 20 Utin N, Wolf My, Wolf Yi, Kunin EV, February 2009. The origins of phagocytosis and eukaryogenesis. Biology Direct. 4, 9. Do. 10.1145/6150-4-9. PMC 2651865. PMID 1924570. 21 Ghosh Dastida U, Gian Guess, Pop D, Robinson RC, July 2015. In Search of the Primordial Actin Filament. Proceedings of the National Academy of Sciences of the United States of America. 112. 39151 do. 10.1073/NAS.1511568112. PMC 4522752. PMID 26178194. 22 Prokaryotic, Kana, Tran, Lin T. Orhant Priu, Magali, Bhaskaran, Johan Dran, Mansa, Edward, Blanchoin. Lauron, Robinson, Robert C. The 18th of August, 2020. Insights into the evolution of regulated actin dynamics via characterization of primitive gelsolin/cafilin proteins from Asgard archaea. NAS 117, 33. 1990419913. Bibcode 2020 NAS 117199040. Bioexif 10.1101/768580. Do 10.1073/NAS.2009167117. PMC 7444086. PMID 327476565. 23 Orsmes N. Kuhn Jr., Jacobs Wagner C., December 2003. The bacterial cytoskeleton, an intermediate filament-like function in cell shape. Cell 115, 6700513 do 10.1016/S0092. 8674, 03, 00935-8. PMID 146755535. S2 SID 144459851. 24 Shy L, Lati, Rothfield L, June 2003. Division site selection in Escherichia coli involves dynamic redistribution of min proteins within coiled structures that extend between the two cell poles. Proceedings of the National Academy of Sciences of the United States of America. 100, 13786570. Bibcode 2003 NAS 100.7865s. Do 10.1073/NAS.12322251000. PMC 164679. PMID 127662229. 25 Luce M, Fischer Friedrich E, Rees J, Cruz K, Schwill P, May 2008. Spatial regulators for bacterial cell division self-organize into surface waves in vitro. Science 320, 5877 78992. Bibcode 2008 SCI 32789L. Do 10.1126/science.11544413. PMID 1846757. S2 SID 27134918. 26 Cock MK, MQCA, Hojcik E, May 2011. BAC-M, an N-terminally processed bactophilin of Myxococcus xanthus, is crucial for proper cell shape. Molecular microbiology 80, 4103151. Do 10.1011/J.1035-2065-2011.07629.x. PMC 3091990. PMID 214144039. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 27 Hena, Tipper DJ, Zhajai D, Hughes C, April 1999. A novel membrane protein influencing cell shape and multicellular swarming of Proteus mirabilis. Journal of Bacteriology. 181, 7 2008 16. Do. 10.1001.18.7.2008-2016.1999. PMC 93611. PMID 100946676. 28 Kuhn J, Brigler, Marshall E, Kant J, Lesser K, Wick S, Jensen G. J, Thurnbickler M, January 2010. Bactophilins, a ubiquitous class of cytoskeletal proteins mediating polar localization of a cell wall. Synthase in Caulobacter crescentus. The EMBO Journal. 29, 232739. Do. 10.1038/embodge.2009.358. PMC 2824468. PMID 199599992. 29 Securo LK, Pincus Z, Gutierrez KD, Byboy J, Stern CA, Volmer W, Salama NR, May 2010. Peptidoglycan cross-linking relaxation promotes Helicobacter pylori's helical shape and stomach colonization. Cell 141, 582233 do. 10.1016/j.cell.2010.03.046. PMC 29205535. PMID 205-10929. Categories: Cell Anatomy, Cytoskeleton. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.